Go, Ariana.
Sharing my screen. Can you see my desktop now?
Yes, wonderful.
Fantastic. Oops, no, that's not, that's the middle of the presentation. All right, here we go.
Great.
All right, so today I'm going to be talking about a genome we've been a paraxylin genome we've been working on for Tuberculosis coracola. Particularly, I'd like to highlight some issues that arose in the post-processing and sort of contaminant screening steps, with the idea that what we encountered might be useful for others also working on genomes from phyla that come from underrepresented phyla in terms of genomic resources. So Priapulida, also known as penis worms, are an exclusively marine phylum of only 22 species currently, but from fossil evidence, it's inferred that they peaked in terms of diversity and dominance in the Cambrian. Priapulins today are still represented in benthic habitats from the tropics to the poles, with species ranging from meo to megafaunal in size. They're found at varying depths from shallow coastal waters up to 5,600 meters beneath the surface. The phylogenetic position of priapulins have been somewhat debated. Morphologically and historically, they've been positioned as part of Scelidophora alongside Chinorhynchs and Loriciferans. However, molecular analyses don't always support this grouping, and here I've highlighted some summary trees from a recent review. As we can see, the position within Ecdysozoa is not at all conclusively determined, and some of the most recent molecular analyses have placed them as the putative sister group to all other ecdysozoans. And this position makes them quite interesting, this putative position makes them quite interesting for investigating the evolution of genes involved in ecdysis, immunity, physiology across the broader group. But to be able to investigate these genes, we need to be able to more robustly determine the position phylogenomically, and with that, more genomic resources are needed. Currently, there is just one publicly available priapulin genome. This is for Priapulus caudatus. It was done out of the McDonald Genome Institute at Washington University School of Medicine a few years ago, back in 2015. It's decent, but somewhat fragmented, especially with this high number of scaffolds and contigs. To help rectify this lack of data for the phylum, we've sequenced and assembled the genome of the meofaunal priapulin Tuberculosis coracola. This specimen that was sequenced for this species was collected from shallow anoxic sediment in Bermuda. Tuberculosis is in a different family from Priapulus caudatus, so this nicely broadens out the sampling for this group. For our assembly, we had both nanopore long reads and Illumina short reads, and we used these to make an initial hybrid assembly using Mazurka, which is the hybrid assembler. This was then polished with short reads using hypo, and then we used blob tools to identify any contaminants that might need to be removed. Further post-processing involves scaffolding, gap filling, and then the purging of any duplicates. The initial assembly had a reasonable contiguity and a decent N50. And through the post-processing steps, this initial assembly was modestly improved, particularly in reducing the number of contigs. All was well until we realized that there was a significant drop in our BUSCO score due to some of the decisions we made in the contaminant screening step. This decrease from around 94% completeness in our initial assembly, which is a fairly good result, down to around 86, mid 80s, which is still decent, but it's not really as gold standard as in the 90s. So this decrease of around 7% sort of warranted further investigation. This was a blob plot we used to identify any putative contaminants. I'm sure many of you are familiar with these, but I just wanted to briefly go over the plot so we can all understand what's going on here. So we have read coverage on the y-axis and then GC content on the x-axis. and blast hits for the contigs in our assembly are colored by the phylum that the blast hits go to. So the greatest number of blobs on this plot are in the no hit category, which is gray. We don't get any hits to the Priapulida phylum. This might be because priapulins are underrepresented in the databases. So instead of hitting to that, the majority of our contigs return no hits as their status. This was just from doing a typical blast, but as mentioned in the discussion following Shoyo's Onakofferin genome talk yesterday, perhaps if we made a database of priapulin ESTs or transcriptomes, we could check that these no hits were indeed priapulin. Next, I'd like to point out the bacterial blobs, which are in red and orange on the plot. This is not entirely unexpected as we're working with a small sample animal. So there could be some like environmental contamination in the DNA extraction step. Thankfully though, like when we removed these contigs and reads from our assembly, there was no impact on Busco score. However, what was unexpected and somewhat concerning is the presence of chordate hits, as we thought this might be human contamination. But when we removed these reads and contigs from our assembly, this is where we saw the significant drop in Busco score, and that sort of warranted further investigation. However, the plot thickened upon a deeper dive into the blast hits file, and we found that all of the chordata gene hits, which are Camelus dromedaeus, or the Arabian camel. Now, this is somewhat peculiar because we don't work on camels in our lab. In fact, no one in our building or facility has been working on camels. Camels are not known to live in the Atlantic Ocean off of Bermuda, and also priapulins are not known to eat large ungulates. So there's something going on here. In the FAQ section of blob tools, they give the following sort of explanation as to why taxonomies of sequences might not be as expected. Assignment of taxonomy is based on a sum of scores. So in the case that there are a number of spurious hits greater than the number of good hits, then the incorrect taxonomy may be assigned. But the question that really begs to be asked in our case is why is this handful of contigs all going to camel? So to try explain what might be happening, together we came up with the following two hypotheses. First, that perhaps it could be due to an inadequate trimming of Illumina adapters in our priapulin genome, but maybe also in the camel genome. And this is sort of causing blast hits to match between them. This phenomenon has sort of been seen in the carp genome as well. There's been some issues with Illumina adapters in that, so we thought maybe the same kind of thing was going on. The second hypothesis that these hits are just due to poorly assembled junk regions, and that's just what's causing these spurious hits between the two genomes. To address the first hypothesis, the first thing we did was run FastQC again on our trim short reads. What we see here is that the Illumina adapters are not present in any significant way. So this sort of makes hypothesis one unlikely. To further address this, we did a long read only assembly for our Tabilicus genome. So this is the blood plot of the assembly, and what we see is that chordata hits are still present and they are all still to camel. And as there were no short reads involved in this assembly at all, it sort of rules out hypothesis one, that the short reads and the Illumina adapters is what's causing this. So to investigate the second hypothesis, that it's junk regions or misassembled regions that are causing these spurious hits, we return to our initial blast hits to take a closer look. And what we found that these blast hits were all to highly conserved genes, such as genes involved in the cell cycle and RNA splicing. In addition, all hits between camel and Tabilicus genomes had significant bit scores and E-scores, which are the measures of sequence similarity. Furthermore, in a blast of the camel genes themselves, it shows that they cluster within other ungulates. So it's unlikely that these are misassemblies in the camel genome, and sort of squashes our second hypothesis about poorly assembled or junk regions. So the next sort of question we asked was, is this just a coincidental relationship between the Tabilicus genome and the Arabian camel genome that's causing these hits? This appears not to be the case. In the velvet worm Epiparapatus genome, which was presented by Shoyo yesterday, there's also hits to the Arabian camel. And in the genome of Priaculus caudatus, which is available on GenBank, we also see hits to highly conserved genes in the camel assembly. So this gives us reason to believe that the issue may not lie with our genomes or assemblies, but maybe something's going on with the camel genome. Now, circling back to our original blob plot, we can see here that the caudata hits in blue cluster in the same area in terms of GC content. Cool. And in terms of GC content with the no hits, while the bacterial hits are not sort of within the same large distribution. And this is also true in terms of read coverage, which we can see here with the bacterial hits off to the side. Due to this difference, we feel more comfortable saying that these bacterial contigs are legitimate contaminants, or like legitimate like exogenous DNA. However, the caudata hits don't fall outside of this no-hit blob distribution in the same way. So it gives us reason to perhaps keep them in our assemblies. So these blob distributions, in addition to our own investigations into the blast hits, sort of leads us to be able to justify the inclusion of these camel contigs in our assembly. And we propose that maybe these camel hits are due to an artifact of the like blast search algorithm or some other feature of the genome. Lastly, when we leave in these caudata hits in our assembly, we no longer see this decrease in Busco score, as we are no longer removing large chunks of conserved genes in the name of quote unquote contamination. And why camel? This remains a mystery and sort of an open question that I would like to have discussed. But perhaps to extend on the words of Sir Arthur Conan Doyle, perhaps it is the villainy within the camel genome with its strange and deceptive ways that is causing these blast hits to occur. And with that, I would like to acknowledge the Museum of Comparative Zoology, my lab mates in the Gerbit lab, and also the GIGA4 team and the research computing groups here at Harvard for all their support and assistance with this project.
Thanks a lot, Ariana, for a very lively and right on time talk. I mean, you managed to fit it in 12 minutes, which is first time since the beginning of the conference, so congrats. And now we have ample time to try to figure out the source of the mystery, which is indeed kind of brain scratching. So I am checking the Keybase channel. There are a few questions there that are all mine, so I will start with this one. First, I was wondering, did you look for these hits in other camel genomes? I mean, there's a bunch of camel genomes, some wild, some domestic ones. If this is really something present in camel, you should find it not just in one species, but in more camel species.
Yeah, so the hits that we got were all specifically to this one genome. However, when we blast the camel genes, like individually, they blast to within other ungulates and within other camels. So I'm not too sure why this particular assembly or genome is attracting these hits from our assemblies. But yeah, like it was only to this one. I wonder if we could constrain the blast hits to not include this one, if some other camel would take its place or some other ungulate. Yeah, you
can do this in BLAST. Even using the online BLAST, you can say, I want to exclude this accession, and then you can see what happens. If there's only one camel, I would have thought maybe it's been sequenced at Karst in Saudi Arabia in the same batch that some swarms, and that would have been a likely explanation. But if you find it other one, then the plot thickens, as Conan Doyle before. Yeah, and Nada is asking a question in the chat. Have you checked how this specific camel genome was assembled?
That is a good thing to do. No, I have not. So we should do that. We should see what was involved there.
Do you know which group sequenced it? Is it Saudi Arabia or?
I'm not sure. I mean, I can see what comes up. But yeah, at the
good mystery plot. That's what
yeah.
you do with GIGA4.
Exactly.
Jean-Francois, I have to say, this is why I encourage Ariana to give this talk here to see if anyone can help with this mystery.
Yeah, no, for sure. Let's see.
I mean, camels are wild, very strange animals in some way. There was a rumor you find in many biology books that they have erythrocytes with nuclei, which is actually completely wrong. They have no nuclei, just like other mammals. But just because the shape of the erythrocyte, it looks like a chicken erythrocyte, people thought they would see a nucleus. So you have a lot of like mirage, Fata Morgana in the literature about camels. So maybe this is another mirage.
Yeah. Yeah, no. Let's see. Where is, oh, here we go. Oh, no, that's, oh.
Yeah, don't hesitate to post the link to this genome in
Yeah.
the base channel or in the chat, and it will trigger also more investigations.
No, yeah, I can post that. That'll be
I
good.
can already foresee myself not sleeping tonight while trying to figure out something. And then, so I had another question also that I posted in the Keybase channel. When you showed your FASTQ plot, FASTQ check plot, I mean, there's still a bit of Illumina adapters. I mean, personally, I would consider it like not yet cleaned. So I was wondering, what tool did you use to trim? Because when I would see something like this, I would actually do some further trimming. It means it's not trimmed properly. I don't think it's the cause of the problem, but that's a bit annoying.
Yeah, no, in the percentages, I think it was about like 2%, like 0.2% of reads that had some like adapter left. I think we used Trim Galore or what do we say? Yeah, Trim Galore is what we used.
Yeah, so I think probably you could change a trimmer. I like a lot skewer, because skewer, even one base of adapter managed to detect it, as it looks for the boundary between the sequence you find in both F1, R1 and R2. So other adapters, you need to find significant similarity with, I mean, other tools need to have significant similarity with the adapter to find it. So you need to have a few bases, like 10, but skewer manages to find even one base, which is quite powerful. And usually if you're running skewer, I get absolutely nothing. And I mean, in the genome we submitted recently to NCBI, the trimming had been done also using Trim Galore, and actually NCBI after asked us to change, I mean, to remove some contigs in which there were hits to vectors. So and then I asked my collaborators, and yeah, they had used Trim Galore as well. So we redid the trimming, and now we are reassembling everything, just because otherwise we'd end up making, like, we have a chromosome scale assembly. If you remove the vector, we make holes in the chromosome scale assembly, which is a bit annoying.
So what was the trimmer that you suggested?
Skewer.
Okay, great.
I can type it in the...
Yeah. Ariana, Chris Laumer is asking you how many specimens you pulled for this.
So I didn't do the wet lab work. That was thanks to Tawana and Bruno, but I believe they used just one specimen and they did a whole genome replication as well to improve the amount of the quantity of DNA.
Yeah, and Tawana is here. She can confirm this, that that's the, you know. So Chris, we were trying to copy you, single specimen, you know.
That's right, guys. I'm sorry I'm not participating more. I'm doing a lot of lab work this month, but I watched and I really enjoyed your talk, Ariana. But yeah, it was one individual whole genome amplified.
There could, in principle, be contamination in the 529 reagents that you used. That does sometimes happen.
Yeah, when dealing with insects, sometimes we see some strange hits as well. And it's actually a contamination by the same bacterium in both, like with, what do you call this? Not mycoplasma, but I mean, very common bacterium in insects. I forgot the name. So if you have the same contamination in two batches, maybe also could come from the kit. Indeed, you have the ketome, as they say, like the DNA you find in many tubes and kits. And maybe it could be something like this. Well,
I did find it interesting, though, that you mentioned a smaller, but a little bit of it in another genome that was done by another group, another preappellate, because we didn't do whole genome amplification for the Nicoffer, for example, right? And just for that tuberculous. So, yeah, that's indeed a big mystery.
So I think it's time now. It's 4:20. Let's move on to the next talk. And the next talk is by, I think, Chris. Is it Chris? No, Claude Power.
Yes.
Thanks. The floor is yours.
So can you see my screen?
Yes, very well.
Perfect, thank you. So hi everyone, and thank you to give me this opportunity to present at the GIGA. And today I will talk about my project that is about identifying stress markers to predict mortality in snow crab. So just briefly, I'm a master's student at University of Moncton in New Brunswick, Canada. And also I'm part of the team of Anne-Marie Dion-Côté, and I'm co-supervised by Simon Amar. So in Canada, in general, snow crab fishery is intense and short because each season that lasts approximately 2 months, each boat receives a quota. And in general, the price of the crab is better at the start of season. So often the fishermen want to catch their quota as soon as possible. So when the resources are there, fish plant can receive huge load of crab every day. That can cause a large pressure on the transformation workers who often need to work 14-hour shift for 6 to 8 weeks. Also, with this short period of work, they usually rely on employment security. Snow crab transformation generally involves to cook and froze the crab. And at the fish plant, the crab can go in 3 lines, going from the most valuable to the less. The first line are the perfect crab. The second line are the less perfect crab. For example, some crab with missing legs. And the last line are the crab that will be shelled. So in general, we could say that snow crab fishery prioritizes quantity over quality. In the lobster industry, they reduce the pressure by keeping the lobster alive in tanks. The tanks allow to stock the lobster until they are transformed, and also allow to sell fresh product later in the year. So the big question here is why there's no such system in the snow crab industry. Well, if we compare the crab to the lobster, snow crab live in very deep water at low temperature. They are a very sternoterm animal, which means they live in a narrow range of temperature. So it's more difficult to reproduce an adequate environment to keep the snow crab alive, but it's not impossible. Indeed, my co-supervisor team have been able to keep snow crab alive in tanks for 6 months. And we think that the major problem here is caused by the transport, because while being transported, crab are exposed to extreme stress, such as handling that can cause physical injury, temperature change, air exposure, decompression, and more. So when the crab arrive to the fish plant, they are already too weak to survive in tanks. We need to change the way we transport the crab to minimize the stress and obtain a crab in better condition. Well, to do so, we need some stress markers to quantify the stress caused by transport. And to obtain stress markers, we need to understand crustacean stress response. So what we know about it? Well, one of the most common stress response in crustacean is hyperglycemia. And hyperglycemia caused by stress is regulated by the crustacean hyperglycemic hormone, commonly called CH8. And in general, CH8 and catecholamine are implicated in the mechanism that controls stress response. But a lot remain to know on their specific function and how they control stress response. For example, CH8 have multiple function and is difficult to relate to stress. To see it in a very simple way, right now it's difficult to distinguish a crab that is stressed from one who is not stressed. The main interest to obtain stress markers is to predict mortality. And we want to quantify the stress because the crabs that are more affected by stress are the ones most likely to die. Yes, researchers have tried to quantify stress caused by transport to have an idea of the vitality of the animal and therefore predict mortality. So in the table here, we can see multiple parameters that can be measured as stress indicator. But most of the time, multiple of those parameters need to be measured to have a global idea of the vitality of the animal. and it's not really efficient. So right now, there is no stress markers that allow to quantify stress efficiently and rapidly to the landing of the crab. That's why my project used a transcriptomic approach to go see the genes that are differently expressed in crab exposed to stress. The main goal here was to find new target as stress indicator and maybe have a better understanding of stress response of snow crab. So how did I get started? Well, I obtained snow crab from local fishermen, and our crab are from Gulf and St. Lawrence in Canada. After I obtained the crab, I placed them in saltwater tank at 2 degrees for a 4-week acclimation. After the acclimation, I divide them in 3 groups. The first group was let in the saltwater tanks as a control group, while the 2 other group was stored at 4 degrees out of the water for 24 hours and 72 hours. After the experiment, I sampled hepatopancreas that I used to extract RNA. Those RNA samples were sent for sequencing. I have been very lucky because while I was doing my project, Seoul National University, I released the snow crab reference genome on NCBI. That saved me a lot of time because I did not have to assemble the transcriptome, and I mapped directly the sequence on the reference genome. After that, I performed a differential gene expression analysis. To first analyze our data, I performed a PCA to obtain a global picture of our experiment and see if the experimental group cluster together. So as you can see here, there's clearly a difference between the 72-hour group compared to the 2 others, and it's more difficult to separate the control group from the 24-hour group. That suggests a more limited transcriptomal response over a short transport time. After the differential expression gene analysis for the 72-hour group compared to the control group, we obtained 550 differently expressed genes. To help to visualize those genes, I made a Volcano plot. So here in blue are the significant genes with a p-value adjusted of 0.05, and I also select gene with the log2 fold chain equal and higher than 4 that you can see in orange. So we can see that the gene implicated in the snow crab stress response are mostly upregulated. So here are the genes that were in orange in the Volcano plot. That means they have a p-adjusted value of 0.05 and a log2 fold chain equal and higher than 4. We think that might be the best stress indicator because the most highly different expressed genes are those who react the most during stress. And just with our first look, we were happy with what we got because we can spot genes that were easily relevant to stress. For example, we got some HOC protein. But you know, we did not do all this transcriptomic approach to find HOC protein. So we looked further to see if we could find some genes that are less well known to play a role in crustacean stress response. And one that seemed very interesting is the tyrosine decarboxylase. So you can keep it in mind, we'll come back to it later. So now that we have a differential expressed gene list, what are we looking for? Who are considered to be good stress markers? Well, we want to quantify stress. So we're looking for something that is low expressed in control group, middle expressed in 24-hour group, and highly expressed in 72-hour group. Something like in the red circle. So the other way is also true because which means we can have something that is very highly expressed in the control group and very low in the 72-hour group. But this is just one part because the biological function is also very important. Because we need something that is easily relevant to stress, but even more important, some metabolites are more easily measurable than others. And if you remember, we're looking for something that is easily measurable at the crab landing. In that case, tyrosine decarboxylase sound very interesting. tyrosine decarboxylase is upregulated in the stress crab, and tyrosine decarboxylase catalyzes the conversion of tyrosine in tyramine, and tyramine is the precursor of octopamine. Why are they interesting? In vertebrate, octopamine and tyramine are biogenic amine that play similar role to adrenaline and noradrenaline of the vertebrate. Like adrenaline and noradrenaline, they have multiple functions. They are involved in regulating a wide range of behavior, physiological variable, and more specifically, metabolic pathway. And that includes stress response in invertebrate. I'm very excited about this because one goal of my project was to find new target. And if we come back to this table, well, tyramine and octopamine are not indicated as stress indicator. Tyramine and octopamine are well studied among invertebrates, but in my reading, I did not read about them as a stress indicator in crustaceans. They were mostly studied for their effect on the behavior of the crustacean and not really on their role on the stress response of crustacean. So we think that tyramine and octopamine are very strong candidates as stress indicators, and we are currently working to measure them in hemolymph by HPCL. Also, for the rest of my gene list in general, we are going to look for some validation. Like I will do metabolomic to see if the metabolite in the hemolymph are consistent with the gene differently expressed in the hepatopancreas. I could also look if those markers are found in other kinds of stress, such as temperature stress, sanity, or sanity stress. So those stress markers should allow to select individuals in the best condition by helping quantify the effect of stress. And here are my acknowledgements, and thank you very much for your listening.
Thanks a lot, Claude, and also for keeping the time. So we have time for questions, either in the Keybase channel or in the discussion here at the bottom of the chat in Zoom. I may have a first question. Did you try to compare the taste of these different crabs at different level of stress? Does it actually make changes in palatability, or it doesn't actually matter for human consumption?
Yeah, that's a good question. We didn't do it, but that's something that my co-supervisors want to do, because the main goal here is to keep snow crab alive in tanks to sell it. So if the taste is not as good as the fresh crab, there will not be interest for that. So that's maybe something we could try in the future.
I'm sure you could recruit some people to do blind tasting among the GIGA community.
Free crab is always fun.
Other questions, maybe?
Claude, I have a question, just related question. Are you guys also trying to do a genome of the crab? And I'm asking that because we're trying to do some long read nanopore genomes of crabs in my lab. And we've been having a lot of difficulties with long read in crustacean genomics. And apparently this is a very general thing for Oxford nanopore. I'm actually looking forward to Juan Antonio's talk later on the spiny lobster genome. But it seems that there's a lot of problem with decapod genome. So I just want to know whether you have any experience with that.
No, it's to assemble the genome, I think, is a bigger project like for a PhD student, and we especially want to find some stress marker. But no, yes, I know in decapod it's very difficult because they have long. I think they have also big transposal elements, so it's that difficult to assemble the genome. But the reference genome I used was like the first genome that was published on NCBI. So yes, I don't know.
I may have some advice on that. I mean, we are doing some genomes of amphipods. It's been really tricky. Even flash frozen individuals would give poor results. Actually, at the end, the best result we got was to the extraction on live individuals. So you can imagine carrying amphipods around live, maybe more difficult for a snow crab. And that I think we tried many kits. The best one that worked was the Kiagen genomic tip 20G. That's the one we are using now for nanopore sequencing of amphipods. So I guess CRAP should be quite.
Yeah, our issues, Jean-Francois, is that we're getting, in theory, good DNA. It just, just doesn't, it clogs the pores, doesn't, so we've tried different fragmentations, different extractions. Apparently, Oxford Nanopore is working on a specific kit for decapod crustaceans because they are losing a big market on people doing decapod genomes because it's not working for some reason that no one has figured out yet. So.
Something that I hear often collaborators saying is some animals work better for nanopore, some other for PacBio, and sometimes it's easier just to shift to the competitor than to try to optimize one particular thing. Like what kills a flow cell, a nanopore flow cell usually doesn't kill a PacBio enzyme and vice versa. So maybe. There was one question in the chat in Keybase from our next speaker. Are there plans in your lab to look at at responses to environmental changes? So I guess climate change issues.
We're not supposed to do this, but yeah, if we got stress marker, well, stress marker could help to give many opportunities for mini studies to help to quantify stress of our. I know, right? I read already the paper that we're checking on looking for stress caused by temperature because snow crab are very stenotherm animal, but yeah, I'm not supposed to do that in my lab, but my project will give many opportunities to for some other researchers to quantify stress, for example.
I see some comment from Kate that she had the same problem with nanopore for tunicates as well. Yeah, there are some tricky animals in the tree of life who seem to have taken side pro or against PacBio or nanopore.
Yeah.
One thing I was wondering also, because you had the assumption that the significant genes would have this kind of temporal increase in response, but have you considered the idea that maybe some genes are only in intense short-term stress, so they will only peak after 24 hours and go down, and some interesting genes are maybe only in the long-term stress, like as we say for human, you have the intense stress, the good stress, and the bad stress. Have you looked into this for your crops?
Yes, I have a comparison for like all the groups. So I have control versus 24-hour group and control versus 72-hour group. So right now I'm really in the analyze of this. So for my presentation, I just focus on maybe those are more interesting, but certainly we can look at those because, we're looking for, like you said, short stress. So maybe those that react mostly in the 24-hour and control group could be the more interesting for us. But for sure, we'll look further on those because right now I'm really in the analyze of all of this.
And I don't know if there's still some comments. I see some comment from Nadej that she's pro both nanopore and PacBio, so they can keep competing to give us better reads for lower price. Yeah, let's keep the competition going. It's healthy, for us actually as end users. And I don't see any further question. There was a comment in Keybase that Simon, I lost my Keybase. Simon Lamar and Anne-Marie are definitively interested in these questions as well, especially from physiological perspectives. We should feel free to reach out to them if you have any ideas of interesting things to do on this interesting model system. So thanks a lot, Claude, for this presentation.
Thank you.
I think we are now ready to move to the next speaker, who is Mathilde Salomon. And Mathilde, when you want, you can share your screen.
Okay, everyone, can you see my screen and hear me well?
Yes, very well.
Okay, perfect. So hello, everyone. Today I will present you some results from my PhD project, which is on population genomics of copepods and gastropods to look at adaptive response to anthropogenic impacts. So freshwater ecosystems are under increasing threats from human activities. In this image, you have a map of temperature increase in lakes worldwide. And as a consequence, populations and species can disperse to new habitats. They can also respond to selection to adapt to their changing environment. And they can also have changes in demography. And all these processes are interacting, and they are important to understand the responses of species to these changes in environment. And they can also be studied using genomics. So the study system that I'm using for my PhD is a lake repod Leptodeatomus manutus and the gastropod Anicola limosa that is found in lakes and rivers. And they are both ecologically important because of their wide distribution, their abundance, and their place in the food web. And both are non-models. So when we started this project, we didn't have any genomic resources. And also, they hadn't been cultured in the lab. And in addition, the copepod is also smaller. So for both species, we plan to do a genome assembly as part of the CanSeq 150 project, and also to apply whole genome pool sequencing. So first, I'm going to present some results for the copepod. So this is the copepod assembly workflow that is still ongoing. So first, we got some 10x genomic sequencing with 2 libraries from 6 and 10 pooled copepods. And we tried to do 10x on the assembly using supernova, but this failed due to not enough coverage. And also we had some contamination issues. So next, we got pool-seq for all of the projects that I will be presenting. And with this, using 2 libraries, I looked at the genome size using Jellyfish and GenomeScope, and it is about 750 megabase, and it is so 2 times less than what we expected from the C-value of the sister species. And also it has high heterozygosity and high repeat content. And unfortunately, we got low coverage for our Illumina libraries. So with this, we try to do Illumina reads-only assembly with MegaHit using 1 or 2 libraries, but this didn't work well at all. So with an assembly size of more than 3 times what we expected and very, very fragmented. So after this, we try to do some Oxford nanopore sequencing, but we had this issue with the pores being clogged and we couldn't get a lot of data, but we still try to do a hybrid assembly, so using 1 or 2 Illumina libraries, and with Platanus Ali, Opera MS, and hybrid spades, but only Opera MS gave us results. It improved a little bit, but still not very satisfying. So for now, we are going to use a reference-free SNP discovery approach with Discno SNP++, and this will be done on the libraries for the different projects. And so using 8 and 14 pooled Illumina libraries, with this method, we were able to obtain 1.5 million SNPs, but after filtering, this number really went down, so I probably still need to play with the filter. So the first project for the copepod is to look at adaptation to historical lake acidification and recovery. So for this, we collected sediment cores in 2 acidified lakes, and we extracted resting eggs from 3 time sample. So before acidification, during acidification, and during recovery. And these eggs were used they were resurrected to do a common garden experiment, and they were also used for sequencing. So we were able to obtain 5 temporal samples, 3 in 1 lake and 2 in another lake. And they were sequenced for pair-ended, with pair-ended. And with this data, I will be looking at potential selective sweep due to the acidification and the recovery with time series analysis. And also, I would like to investigate demographic effects. So the next project is to look at the local adaptation to climate. So for this, I have a latitudinal gradients. There is 3 regions in Quebec, Canada, and 1 region in Greenland from collaborators. And this gives us a total of 8 populations, and we sequenced 200 individuals per pool. And with this DiscoSNP software, we were able to get 120,000 SNPs. And with this dataset, I am looking at the population structure. I am planning to do environmental association analysis, so in part with the especially for the climate, but also to other environmental parameters, and also to investigate the demographic history. And for this, I will use climate databases, and I also have access to physico-chemical characteristics that were collected during sampling. So these are preliminary results of this project. So in this PCA, we can see that samples that are close together in the gradient are also clustering close together. That's a good results. But the populations from Quebec are not clustering according to the latitudinal gradient. And this may be due to the demographic history, because this species had a range expansion after the last glaciation that went from south to north, but also from west to east. So that could explain this pattern. And also the Greenland samples are highly differentiated from the rest and between them. And we think that this may be due to genetic drift because of small population size, as these populations are at the range edge of the species. So looking at localization with PC-ADAPT, we got 555 outliers, which represent less than 1% of the dataset. So this will need to be investigated further. And we also were able to calculate average pairwise FST, which is pretty high, but that's what we would expect from such a large sampling, and also because we expect that the species has low dispersal abilities. So the final project for the GIGA4 is to look at macrogeographic adaptation in a connected landscape. So we are changing scale for this project. It's in Cape Race, Newfoundland in Canada, and we sequence 14 populations. And with this data, I will look at population structure, do environmental association analysis, especially looking at the summer temperature and the pH, as there is a wide variation. But also to other parameters that we have measured. And I will look at demography in this system, because there is some important differences in population size between the samples. So next, moving on to the gastropod, I am interested to look at the local adaptation within a dynamic river ecosystem. So this study takes place in the St. Lawrence River around Montreal in Canada. And this river has been invaded by the round goby for about 14 years. And the goby is a predator of this species that we are studying, Anicola limosa. And it has had important impact on the demography of the species. And the other interesting aspect of this study site is that there is a spatial gradient of invasive predator impact and calcium concentration. So in the St. Lawrence River here, there are goby present and there is high calcium, but in the Ottawa River here, there is no goby and the calcium concentration is low. And there are also other environmental parameters of interest for the species that are varying. So the dissolved organic carbon, as well as the total phosphorus. So we sequenced 12 populations, 6 in each part of the gradient, with 14 individuals per pulse. And de novo 12 genome had been obtained before I started this project using 10x sequencing on one individual, and it was assembled with supernova. So the genome size is about 2 gigabase, and the genome is still pretty fragmented it's pretty fragmented. And but with this genome, we were still able to get about 50 million biallelic SNPs. So first, looking at the population structure, the populations are not structured based on habitat types. So this is contrary to what we expected if there was isolation by environment. And also this population is isolated from the rest and is the only one that has goby present and low calcium, so the parameters are inverted. And we also found low pairwise FST, so there is strong gene flow in this system, and we did not expect that because this species deposit the eggs on the substrates, and the eggs and the larvae are not released in the water column. We also looked at the demography, comparing the diversity indices for the populations from the 2 habitat types, so in gray and blue here. And we found no difference in these indices for pi, theta, or Tajimas-D. But for Tajimas-D, we found that it was slightly negative, and this could be explained by recent expansion after bottleneck. So I will investigate this further using demographic models. And finally, we did genome scans using bypass and other methods. So the interesting aspect of bypass is that it takes into account the demographic history. And with this analysis, we were able to find about 130,000 SNPs. And that were significantly associated to one or more covariable, and this represents about 1% of the data set. And also it gives us a regression coefficient of the association between the SNP allele frequency and the different environmental factors that we tested. And we found positive and negative association. So to summarize, we found signals of selection and also potentially local adaptation, as we found some overlapping SNPs between the invasion and also the other parameters of interest. So the preliminary conclusions for this project with the gastropod is that for the population genetic structure, we found important gene flow between the populations of the 2 habitats. For the demography, we think that there was potentially a mild bottleneck because of the predator impact. We found signatures of selection from the predation pressure, the calcium limitation, and the resource type or abundance, as well as local adaptation. And the next step for the study will be to do functional analysis. So we don't have an annotated genome for this species, and at this point we cannot annotate it ourselves. So I am planning to use the annotated genome from another gastropod species. To summarize for the overall project, this will bring new knowledge of adaptive responses of freshwater species to multiple environmental and biotic pressures. It will also serve as a baseline for future ecological genomic studies of freshwater copepods and gastropods. And unfortunately, the genome assembly has been challenging for the copepods due in part to genome properties, but also to technological issues. So I would like to thank you for listening to my talk. I would like to thank also the Barrett Lab, the Dairy Lab, as well as our collaborator from the McGill Genomics Center, our collaborators from the Switzerland, and our funding sources. And I will be happy to take any questions.
Thanks a lot, Mathilde. You managed to cover a lot of ground in the allocated time, so it's really nice. So we have
this is challenging.
So you can ask questions in the chat of Zoom or in the Keybase folder. I may have one thing to start with. I noticed that you use, or you tried in some times, or it worked in other time to use linked rates like 10x to generate your reference genome sequence. And that's something we haven't even much talked about before in the conference. People usually go from nanopore, PacBio, it's kind of a big divide, as you can see. So what could you say about the reason why you chose the 10x? What are the pros and cons? Would you say it's a good way to do a reference genome?
So I think we started with 10x because it became very popular when we started this project, and then Oxford nanopore also took over. And the reason why we switched is because we didn't have good results with the 10x, probably because of this contamination issue. So we are working on that. But I think at this point we don't have enough coverage, either for the long reads or for the short reads, to get a good assembly. So
there was one genome for which your final reference was from 10x, right?
Sorry, for what?
One of your genomes, toward the end of your presentation, you had a reference genome based
Yes.
on 10x.
Yes, so the gastropod, yeah, we didn't have the same issues. There was more like material to start with. It's a bigger species, and yeah, like using the same method with supernova, like there was no issues. Well, the genome is fragmented, but yeah.
In terms of costs and in terms of computation, was it cheap? How was it?
It's been, yeah, both have been pretty expensive. So all this represents months of work on the cluster, and we've spent a lot of money on the 10x and the nanopore, and for now it's not working. So yeah, but it's still data that we can use in the future when we get maybe better data, or it's not lost data, but
yeah, I would like to stress, like I mentioned before in some discussion, graph and zip, which is a problem we've been working on in my group, to phase assemblies using long reads and high C, but now we can also use 10x, I mean, linked reads to phase assemblies. So, I mean, you may want to keep this data, because maybe at some point it can help you to resolve your deployed assembly.
Yeah, that's what I'm hoping. Like, so I tried to do the hybrid assembly as much as I could, but there was no way to integrate the 10x in any of the hybrid assemblers to date.
So using graph and zip, you need to have a GFA. You need to make sure you do a preliminary assembly that returns a GFA, and then you can map the linked read on the GFA and resolve the haplotype using this. Yeah. But
yeah, maybe that could work. Yeah.
We still have a bit more time for questions. Other comments? Or look at the Keybase channel. There's no other question there. Yeah, no, I mean, I was quite impressed to see. So it's for your PhD thesis, right?
Yes.
So it's a lot of things for PhD thesis.
Yeah, so I think what's really adding, and that's the part I didn't do, is so the wet work and the sequence, the wet lab work and the assembly for the gastropod. I didn't do that. They gave me the pool sequences, and I just did the downstream analysis. But yeah, it was a bit challenging that most of my PhD was based on this copypod assembly, and it's not working. But at least there is like a way to get some data. It's not a lot, but yeah.
I'm happy that Disco SNP worked for you. I tried it on a rotifer project. We never managed to get something good out of it. I wonder, do you know the heterozygosity of your organism? Because then maybe our problem is the rotifers is maybe too heterozygous. Oh yeah,
it is very high. Yeah, it's high. But yeah, it works well. And this data I haven't even tweaked with the parameters of Disco SNP, so maybe I can even improve this. But the downside is that we don't know where these markers are, like if they are really representative as the genome, and yeah, of course we cannot do functional analysis. So yeah, there are some limitations, but
sounds tricky. But I mean, I'm impressed it worked so well for you. So congratulations. I mean, we broke our head on Disco SNP for weeks and didn't get anything out of it. So it's very nice. Any further comment or questions to Mathilde? Otherwise, I think it's 59. I think it's time to move to our next speaker. Thanks a lot, Mathilde. And our next speaker is Juan Antonio Baeza.
Yes, how are you doing today? Can you see my screen just to make sure?
Yes, it's perfect.
Yeah, thank you. Thank you very much, Jean-François, again, to give me the opportunity. Antonio Baeza from Team Nanopore, not really here. And I want to talk a little about the utility of low pass. I'm very interested in low pass, relatively cheap nanopore long-read sequencing for mitophylogenomics and barcoding research. And for that, I use in a sort of 2 or 3 actually iconic marine organisms. Have been working with spiny lobsters, sharks and shrimps right now, but of course I will only be speaking about spine lobsters this time. Yes, because of the venue, right? Sure, you know better than me that the short fragments of the DNA has been markers of choice for the last 20, 30 years, together or without nuclear markers, to reveal phylogeographic population genetics and phylogenetic relationships, right? Of course, we have SNPs and rat sequencing nowadays. However, most recently, whole entire mitochondrial genomes are quickly becoming markers of choice, again, to reveal phylogenetic, to accomplish the same task, phylogeography or phylogenetic relationship among closely related organisms, in addition or without, again, SNPs coming from rat sequencing or genome, or low-pass genome sequencing. The issue here, and my question is much more pragmatical, less theoretical, but I'm very interested in retrieving these whole mitochondrial markers very quickly in time, because I want to use them to understand mislabeling in the industry or illegal trading of shark fins with colleagues in Central and South America, or also mislabeling in lobsters. That is commonplace actually, but poorly reported. And right now, the gold standard for the generation of these complete mitochondrial genomes is short-read sequencing, thanks to the Illumina platform. But again, the things are relatively long, from start of sampling of a specimen all the way to sequencing and assembling the mitochondrial genome. It can take several months, up to a year of a couple of years. And that's why it's very interesting now to have the opportunity to work with long-read sequencings from Oxford Nanopore on PacBio. And you can already see the differences here in terms of size of the tools or equipment that we use. Depending on which team you are, right, I will argue that PacBio is ultra, not even ultra, it's uber expensive. Yeah. The device itself is thousands of dollars, and I'm working a small, medium size, I will argue small lab, and I usually interact with people that doesn't have too many resources for sequencing. So PacBio really is not an option compared to Nanopore. Reactants are relatively cheap. The device itself is small. The library construction is relatively cheaper compared to PacBio. But we know that both platforms have issues with initial error rate on the sequencing, right? And yes, I agree that PacBio is improving the initial error rate with the hi-fi protocol nowadays, so sort of almost solved. Oxford Nanopore is lagging behind, but as we speak, sure, you know that you have the Q20 experimental chemistry that apparently is claiming can improve error rate very highly. Let's see if that happens, right? Still, I will argue, if you're interested in the quick retrieval of genomic markers, and by that I mean full mitochondrial genomes, Oxford Nanopore is the way to go. You can take the device in situ and is much cheaper than PacBio. But we have this problem with initial sequencing error rate. So with that information in our minds, what I'm interested in doing here is testing whether or not mitochondrial genomes can be sequenced from low coverage, so relatively cheap sequencing, long-read nanopore sequencing, exclusively. So we are aware bacterial genomes, even human genomes, have been generated using high coverage nanopore sequencing, but there's little benchmarking about the error rate of those genomes, right? Other than a couple of papers coming recently. And most frequently you use, in parallel with nanopore, you use Illumina sequencing for correcting. So again, I'm only interested in using nanopore exclusively sequencing for generating these genomes. And then something that is not very commonly reported, I'm interested in checking the accuracy of these long-read assembled genomes. And in this study, I will do that by be benchmarking these long-read assemblies with a short-read gold standard reference mitogenome from Illumina sequencing. And last and not least, regardless of the accuracy, I will have news for you in short, right? I want to know if whether or not these assemblies are accurate or not. I want to test they are reliable enough to conduct mitophylogenomics and barcoding, just because, again, quick analysis focusing on mitophylogenomics and barcoding with this long-read assembly, accurate or not, will permit us to tackle these issues of mislabeling of these species in the trade. So for that, I use, or I will mostly speak in this case, not necessarily about silky sharks. I have some data on the silky sharks, but I will be using the Caribbean spiny lobster. Why? The most valuable from an economical point of view fishery in the Caribbean. There's plenty of artisanal and industrial fleets targeting this species. All populations are fully exploited or overexploited. There's problems with this species in the natural environment. There's plenty of supply chain mislabeling in the species. We're interested in investigating that. And of course, these are very ecologically important species. We want to know what happens if these species experience drastic decreases in population, how coral reefs actually, where they live, are affected in terms of community membership abundance. So let me tell you first about testing or whether or not complete mitochondrial genomes can be sequenced from low coverage long-read nanopore sequencing data exclusively. The first thing that I did do was I generated what I call a gold standard reference mitochondrial genome using 0.5 gigas of 150 per short reads Illumina platform. I was able to generate this genome with a high coverage of between 7 and 830 X coverage. I used two assemblers, Get Organelle or Novoplasty are the most recent ones that are able to have really good quality and assemblies. If I annotate these genomes, Illumina genomes with Mythos and Mythos Zoo, they have the same synteny that is conserved in the genus Panilurus in lobsters. There's no issue with stop codons interrupting the open reading frame. So these are reliable, high quality, fully accurate Illumina genomes. And this is going to be the one that I will be using for benchmarking the long read assemblies. This workflow, I don't like to use pipeline, the workflow for assembling the long read genomes with the nanopore. I was interested in low coverage. So what I did here is I generate a library using regular protocols, and I sequence that library in a single cartridge or cell together with 4 other libraries, right? And that was that gave me a total of a little less than 78,000 reads of different lengths, right? I trimmed those reads with pork chop. I quality check them and eliminate low quality reads with FastP. And then I use 3 different strategies for attempting the assembling of these mitochondrial genomes: Fly, Unicycler, and Revaler. Fly and Unicycler are the node assemblies. Revaler is a reference-based assembly. I use different strategies. In the case of Fly 1, 5, and 10 polishing rounds after assembly. In the case of Unicycler, I use bold, normal, and conservative. Apologies, because of time, I will not be able to tell you too much about the details of these assemblies. And in the case of Revaler, I use as references, I use or P. argus, or the Caribbean spiny lobster, or other species that are more distantly related. So I was interested in checking the effect of how related is the reference to the reads in terms of the accuracy of the resulting assembly. After assembling, I did a little trick here. I'm interested, as I told you here, in generating quickly these genomes. So I inspected the excuse me, I'm forgetting the word. I inspected visually the assembly graph with the software Bandage, and specifically I look for a circular assembly that should be approximately 8,000 base pairs. And then I blast that assembly, and that allowed me to determine if indeed that circular assembly was indeed a mitochondrial genome or not. And last and not least, I use MEDACA as an extrapolation. The base calling, this was conducted a couple of years ago. The base calling was at that time conducted not with GAPI. GAPI wasn't performing very well. Right now it's doing very well, of course, but GAPI at that time wasn't performing very well. Still, this was a little experimental, and used MEDACA for extrapolation, even though I didn't use GAPI. And spoiler alert, does it work if you don't use the exact model? Yes, it does work anyways, and I will show you that in yet. And use again the Illumina reference genome. I explore completeness, length, coverage, and identity patristic distance as measures of assembly accuracy. This table shows you the number of contigs and the type of contigs that I got using the different assemblies, de novo or reference base, the different strategies, and the use or not of MEDACA. And the most important information here is that with all these assemblers, we were able to retrieve circular mitochondrial genomes. The blast indicated that they were indeed Panirurus argus on the one hand. The length of them was a little different. The coverage was relatively good, 30x or above. And the patristic distance indicates that they are not identical, but they are very similar. And that is related to the total number of errors that we obtain. And I will show you in brief immediately here the analysis of type of errors that we obtain with our different assemblies. But the bottom line here in this slide is that to the question, are we able to assemble mitochondrial genomes using low pass nanopore sequencing? Yes, we're able to do that, and all of the assemblies are circular. I want to comment also that I did use Cano, but Cano actually is known to have issues with plasmids or circular assemblies. Cano didn't work. That's why I'm not presenting information about that assembly in this case. Let me show you an analysis of sequence errors in our different assemblies, strategies, and with and without extrapolation with MEDACA. There's two main points here, this number of sequence errors in this graph, again, for the different assemblies and strategies. But what I want to highlight is that the bars go up and down, up and down, up and down here. And that is the effect of extrapolation with MEDACA. So again, MEDACA does work. Sometimes it improves the assembly accuracy by 20–25% on the one hand. And the other main point to take from this graph is related to the dominance of the color, of the deep green color. And that means, again, that the most common error that we observed here was a 1 base pair homopolymer deletion. That is an issue known to happen with the nanopore assemblies, but it has been rarely really reported and quantified. So this is one of the few studies that actually is reporting this. There's a couple of studies doing the same with bacteria, but these are very detailed error description, I would argue. So long-read genome accuracy is high, I would argue, but it's not full, it's not perfect, right? Okay, so it's sort of a bummer, but the point is, okay, can we use these assemblies for a different type of studies, right? So this is a set of the long-read assemblies after annotation with MITOS and MITOS2, yeah? And what is interesting on top here, I have the reference genome assembly with Illumina, and everything is black are genes that when they are annotated in MITOS and MITOS2s, they are indicated as interrupted because of the presence of stop codons in the open reading frame. So MITOS and MITOS2 report them as duplicated genes. That is not really the case. The point is that in some of these genes, the number of stop codons is so many that the annotation tool is telling you that they are duplicated on the one hand. So that is not look from that side. In some cases, even the problem of stop codons in the open reading flame was such that the in silico tool tell you that the gene wasn't there, but if you manually check the sequence, it is there. Just the problem of stop codons so much produced by the, again, homopolymer error in these assemblies that is producing this issue with the annotation. So to the question, can we use these assemblies, low pass, long read assemblies with the nanopore, for instance, to study selective pressures in protein coding genes? The answer is no, right? We're using, with some colleagues, we're working on the effect of behavioral traits or social structure, sociality versus no sociality on selective pressures in mitochondrial genomes. We're doing that with Illumina because we know now that we cannot do that with a nanopore, at least. Still, although highly accurate, these errors are precluding generating reliable annotations with MITOS and MITOS2. And the last question was, can we use these assemblies even though they are not perfect for mitophylogenomics and barcoding research? So to answer this question, I have two analyses here. What I did first is taking from GenBank all complete mitochondrial genomes on the infraorder Achellata of decapod crustaceans, all of them available. And I use all the nanopore assemblies that were imperfect, plus the perfect, fully accurate Illumina genome to do this mitophylogenomic research. This is a maximal likelihood phylogenetic tree with a total of 42 terminals. I use 13 protein coding genes, nucleotides, but I use a second analysis using only amino acids. It's giving me the same results. The bottom line here is that in this phylogenetic tree, all the long read assemblies plus the short read assemblies of P. argus congregated are clustered into a single fully support, thanks Jean-Francois, phylogenetic tree. So yes, we can use it for phylogenetics. This is a second barcoding analysis, a 500 nucleotide segment of the COX-1 gene using 1972 terminals, 1,972 terminals. And again, we have a very clear fully support cluster for Panilurus argus, and within that cluster, we have a fully support cluster for all these long read assemblies. So, although not completely accurate, long read mitochondrial genomes can reliably identify the sequence specimen as belonging to the species that we're working to, and it can differentiate the specimen from other closely related and distantly related species in the same genus, family, and order. Bottom line, yes, we can use this to explore mislabeling, for instance, in the trade industry. The idea here is, if you allow me, Jean-Francois, last 30 seconds, right? This is a proof of concept. Again, we want to use, we believe, or in my life, we believe that nanopore is a disruptive technology. Unfortunately for us, PacBio is not, but we can change the research scenario. We're interested in transferring, bringing in situ these devices and explore mislabeling, explore illegal trading in Central America, in South America with colleagues. So to avoid parachuting science, we want to transfer pipelines, workflows, and the device itself. And also, of course, this will help decrease the cost of exploring metapopulation connectivity too in this Caribbean spiny lobster. Any detail that I forget, this is a paper recently published again, and thank you so much for your time. I'm not sure if I have a time for questions, but thanks for Jean-Francois. I appreciate for that.
Thanks, Antonio. Well, I mean, we have time for maybe 2 minutes of questions. Anyone? Otherwise, I have some already in the Keybase channel. First, maybe a first quick question. I mean, what about Flangel? Have you tried, you showed a regular flow cell, maybe Flangel would be better for your intended use?
You're right, so that's a good point. haven't tried. Before the pandemic, I was planning on having several trips to Brazil and Costa Rica, actually, with a flunch. It didn't happen, but you're right. That's something. Of course, we will expect a smaller number of reads, but I will argue we will have enough against to relatively quickly be able to extract information that will allow us to determine illegal trading or misleading in the industry. So you're right. That's something to totally to do.
There's one question from Jeff Robinson in the Zoom chat, wondering about the reliability of the nanopore device. Did all your runs run properly without any issue? Or did you have some failure in some runs?
Yes. So as we speak, we're having some runs. I think it's a matter of getting to know the tricks, So I still don't get the total number of reads that nanopore claims you can have from a single run, right? We have solved the issue of bubbles, right? If you get a bubble into the cell, that's it, right? Not really that's it, but that will really decrease your total output, right? But I think we hope that in the future, with more practice, we're able to get the tricks properly work, so to improve outputs. But again, I have the impression that is get the money and buy the cells and mess up things until they work, I guess.
And another question I had is whether you had tried to figure out the percentage of your reads that come from actual mitochondrial genome, because there's a preprint published, I put the reference in Keybase, that show that the mitochondrial genome is less represented in long reads compared with Illumina reads, because simply, it's a short bit of DNA, so most protocols select for long reads. As a result, you decrease the amount of mitochondrial DNA. Do you have an estimate of your percentage of mitochondrial read?
No, that's a good point. I don't, but that's a good point. So we fragment to, I don't remember. It was a couple of years ago, but we were very interested in fragmenting anywhere while preparing the library. Just one, not to clog the pore, right? And two, to have some representation. I think that's something that with the twisting the protocol, you should have no problem, actually. It will be about having the right fragmentation working out for the specific length of the genome that you're interested in. But you're right, that's a very good point to keep doing research on that. Thank you.
Because you may have a higher representation of nums as a result if you keep your long read. Right, we're already 2 minutes late, so I think we will close the session right now. You have some further questions in the Keybase channel. Antonio, please go on and answer them yet. Now we have a little coffee pause where we can head to the Gazotown if you want to have a little more stroll in this virtual sceneries. And we reconvene in 18 minutes at 5:40 CET for the very last session of the meeting. This is the General Assembly. So please try all to be there for this exciting event. And we'll take a group picture at that time. So it would be nice if you are all represented. We'll start with a group picture, actually. Thank you, and see you in a few minutes. So welcome to the very last session of GIGA. And that will be our business meeting / General Assembly. Thanks for being here for this nice event. And I pass the word to Joe, our president.
Thanks, Jean-Francois. Yes, thanks for everybody who helped put this together, and yourself and the committee and the attendees. And thanks for the PI people who purchased a registration fee. It was nominal and voluntary for the most part. We wanted to early career researchers to attend, and I think the count currently from Kevin was about, I saw the first couple days we were up to 60 to 70 at several sessions. Fewer today, about 49. And then here at the General Assembly, the Alliance, thanks for sticking around for this. But Jean-Francois can let me share the screen. Here we go. Just as we go
Is it only me, or I mean, the sound is very choppy on my side?
Um, this is- Can you hear? Can you hear my screen? Arby, can you see my screen?
I don't see anything. It's like it's about to freeze on my
List
side.
of items. Hopefully, Francois will come back. So basically, this is my last address as GIGA president, and I've really enjoyed it. It's been great watching and seeing this group grow and mature since we first started in 2013. And just a testimony, this is our 4th conference, and hopefully we'll have the 5th one in person. If we can get the virus under control. Overall, though, this has been great to have it online and just to keep us going. And I hope we continue to grow. We are the first agenda here is that we did obtain some funding from the Gordon and Betty Moore Foundation, and this is going to go into a fund to push our international standing in terms of we're going to see if the funding can go to underserved nations, most notably in the global south this coming year. We obtained this from the Moore Foundation, about $14,000, and we're going to work with the next administration. The next board is going to develop a new set of fellowships, which can go out next year. And again, the funding status we have now is we are nonprofit, not for profit, but we aren't tax exempt. So any income we have is currently being taxed, and we just haven't gotten around to getting status. We can do that in the U.S. by applying for 501c3. This gets a little bit involved legally, so that's why we have been hesitating. But when we get funds from Moore Foundation, they require us to be a public charity. So that's something to consider. The next board to consider is that that we become tax exempt, because we can't accept larger donations that way, or we can once we are with this status. So if the membership agrees to this, and there are going to be some amendments that are going to be posted for membership voting and acceptance, this will possibly be one of those. So I think that would help us overall to grow further, and grow our fellowships and our mission. Now, I'm not sure if Kevin's on the line, but if not, he will post proposed amendments. Right now, they're fairly modest and brief, mainly addressing our elections, which we noticed needed clarification. So the bylaws, again, are on our homepage. And if any member wants to make any suggestions to make the bylaws better, more equitable, feel free to send suggestions and new language to the board, and they can make those changes and bring them up for another vote. I think we can amend that as we like. As a reminder, GIGA was meant to be a bottom-up grassroots community, and so far it's been like that. We're trying to. The board is just there to coordinate ideas and initiatives. So, and then disseminate that to the rest of the community or society as we are. Then a suggestion I had, because I attended a African Biogenome Project meeting recently, was very encouraged to see more groups that want to do large-scale genomics. This is an offshoot of the Earth Biogenome Project, which we are a community of. We're a network of that network, so we can continue to work with them. They are a much bigger group, an umbrella, and hopefully the next board will also continue to interface with that group. But what they do is have regional representatives, and I think to grow GIGA as well, with the invertebrate focus, we could start welcoming regional representatives or ambassadors, so to speak. So the African BP has already agreed to having a representative, and I'm not sure if is Joseph online. Were you part of that meeting? He was here earlier, and I'm not sure if he's still on the line. But Joseph Karingwa, were you there?
Yes, I'm still around. Yes.
Are you in the African BP group?
Yes, I'm on the committee in the Africa BP.
Great. So, if I talk to Thank God, and I think that's a something that if you'd like to join us, or we can have a representative that you want to send to our regular meetings. I think Thank God was open to that, and if you're open to that, I think it would be nice to have that dialogue and to spread resources and help other biologists in your area. Do you have any comments on the BP project?
Yeah, so far I think the consortium is growing, and we have already started sequencing with 2 species from South Africa, genomes, and it's a growing community and still building collaboration with this big consortium like ALGA and all these other Tree of Life project, the Earth Biogenome project, and so also now GIGA. It's really growing and exciting, and it's all about conservation, agricultural production using genomics in Africa.
Excellent. That's great. So glad you attended, and I think you should be invited or attend any of the meetings going forward, and have a representative from your group, and then also from other continents and countries to make us international. Thank you.
I see one hand raised.
Good. Did you have a comment? Yeah.
Yes, I think it's a fantastic idea, Joe. And we are currently or have already formed the European Reference Genome Atlas initiative. Jean-Francois is also deeply involved, and Joseph also, because he's in Phil Schiffer's lab. And there are, I think, more than 350 participants already in the ERGA project, and we have a large pilot study. Joseph also knows about the pilot study, where we sequence local species in the different European countries. It's a very long list. And this initiative is to a large part comes from the vertebrate genome project, but we put the hands up for the invertebrates, of course. So I think it would also be good if GIGA is on the list. It's mentioned as a partner on the ERGA website. It's it's erga-biodiversity.eu. I think GIGA is mentioned there already as a partner, but I think it would also be good if the president or the next president could reach out to the ERGA Council or the ERGA chairs for a more formal collaboration. Could even be invited to one of the plenaries or council meetings, but probably plenary meetings. I think the African Biogenome Project was already invited there quite a while ago, but I think it makes sense to reach out to other regions and continents should there be any regional initiative.
Yes, the ERGA initiative is really exciting. So thanks for that, Kurt. We'll definitely keep close contact with that. It's benefit for all. So before I go on to the next item there on the homepage, I see that Kate and Abda are online still, and I just wanted to, acknowledge them as they were recipients of our first fellowships, and thanks for entering those competitions and doing your work. Did you all want to say anything regarding your projects or any updates? I'd asked Abda, she wanted to give a quick, and then Kate, she was also agreed to a blog as well. Abba, sorry.
Yeah, could we start with Kate because she's the first winner, like she won before me.
Sure. I didn't give her much notice, but
that's okay. Sorry, I'm just moving spots because it was a little loud where I was.
You got to eat a lap tour, it's nice.
Hi, yes, my name's Kate. Thank you so much for the GIGA scholarship. I just finished my PhD, but I received it during my PhD where I was working on salbutamol. And the funds really helped me work and finish up the genome for salbutamol and also for another salp species, Salpa aspera. So that was really, really, really helpful and informative for the species, and hoping those papers will be out in the next few months. So stay posted. And thank you again very, very much.
yeah, thanks for a great presentation last year at the Biodiversity Genomics Conference.
Thank you for having me. Hopefully I'll be back.
Yes, back in your postdoc. Do you have a postdoc yet lined up?
Yeah, I actually just started my postdoc 2 months ago. I'm at the Gloucester Marine Genomics Institute in Gloucester, Massachusetts. And I'm focusing on studying aging and longevity in the long-lived sea urchin, which lives for about 200 years.
Sounds very exciting.
Thank you.
Abad, do you also have some comments or news you would like to share?
Yes, I have a short presentation just to give you an idea of what I'm going to do. I've got the GIGA Ignite inaugural fellowship, and I still have it. I still have to start the project, so I'll just tell you a little bit about it. So my project is regarding whole genome sequencing of a sponge, Senecarella cf cavernosa. This is the sponge. I have successfully reared it in my host lab in Goa, India. And it's a very hardy sponge, I would say. Like it responds very well to lab conditions, and I have already done my PhD work on it. I recently submitted my thesis, so I've worked on regeneration in the sponge and spatial competition. And for this GIGA Ignite Fellowship, I'll be working to generate a chromosome scale level chromosome scale whole genome assembly of this sponge. There'll be 5 sub objectives in that. One is genome size assessment using Fulgent and DNA densitometry. Second one is long read sequencing using PacBio. That will be done. That will be outsourced here in India. And then there's high-C sequencing with the library preparation will be done in Professor Float's lab in Brussels, and the sequencing again will be outsourced. And then there's genome assembly using one of these programs I've been hearing about, and then we'll be using these long read assembly and make them into complete chromosome sequences using high-C data with graph unzip. And then the last part is, because this sponge is not yet completely confirmedly identified, we'll be comparing the COI or 18S markers with the published markers, and then hopefully identify this sponge. And these are some of the related publications. The first two are my publications of the transcriptome of the sponge, and the other two are related to techniques. Thank you.
That's great, Abba. Sounds exciting. And we need to compare notes because our lab's doing Cinecarella alicata.
Yeah.
And we just, I don't know if you've seen your bioarchive. We have now published, it's in press, the transcript of alicata, and also the Sanger Institute's doing the genome as well. So I think to compare with your species.
Yeah, that would be great. I actually talked to one of your dissertation students in a conference last year, and I got some information from him regarding what you're doing in the lab. That time, I think the genome wasn't sequenced yet, so we can definitely compare notes now.
Yes, it's in the pipeline, so I'm not sure exactly when, but the ASG project, as a segue, is doing 50 invertebrates and symbiotic invertebrates for a photosynthetic focus, and then another 450 for a total of 500 total invertebrate species times 2. So we're looking forward to that. The latest update was that, and Chris was on this call, that they're having issues with some extraction protocol, and it turned out the sponges are turning out the best in terms of the best DNA yields. And so this is a topic we want to talk about in the open forum here in a few minutes, DNA extractions. Next item I have, actually is going to insert this as well, is that Gonzalo, our treasurer, has a report on what our current status is, financial status.
Yes, briefly, since the last meeting, if you remember, everything was deposited in a PayPal account that was mostly managed by Kevin, I think Kevin Cossett, and we, once I became treasurer, we created a bank account on an American bank called TD Bank, which is basically an online banking system. We wanted to do something that, you know, all that we could transfer easily the account from one person to another. Right now, Joe and I are the account holders. We'll need to switch that to the next president. But basically, we put all the money in that account, and then we started as a non-for-profit organization, as Joe mentioned. So we needed to file taxes because now we were an entity, and obviously, all the money that we carried into that account counted as our initial income for the society, and we were liable for taxes on that income. So basically, we started that account with about $7,000. This year we needed to pay taxes. We paid off the fellowships, and our current account has $3,033. This is not counting the money that people have paid for registration into the PayPal account. So we need to transfer that into our current account. And then hopefully, as Joe said, we're now looking into becoming a non-for-profit, which means that we won't have to pay taxes on income in the future. That's the main reason, especially for large gifts like the Moore Foundation. So we can put all that money to use for especially student fellowships and things like that. You know, because we are, as Joe said, an open society. If anyone has any questions about the accounts, I mean, I have everything to the send in a share folder with other GIGA or with the you know, the whole GIGA executive board. But you know, we're okay. We have some money. We don't have a lot, so this is when Joe now is going to step in. Once he steps down as president, he will step in as the person leading the fundraising for the society. He's been very active in that way, and I want to thank Joe for being able to bring money to GIGA.
Thanks, Gonzalo. We'll try. And thanks for holding it and taking care of it. Hopefully we'll be successful with fundraising going forward. So next is, you might have noticed that the homepage got a facelift a bit, and we had a few issues with the URL, but I think that's fixed now. And our very competent webmaster, Jeff Robinson, has been in charge of that, and it's a lot of work. So we really appreciate this, Jeff. Do you have any comments?
Yep, sure. Just to say that I'm still learning the system. So when I started doing it, we had Joomla, and then we switched to WordPress, and now we have WordPress, but we had just recently gone from our previous service provider, ICDsoft, and migrated it to the Google Cloud. So at each of those steps, I'm kind of learning, you know, new things about web development. But I would say that now that we're on the Google Cloud, we have a lot more flexibility and capability to integrate data and data analytics seamlessly into the website, a lot more than we did before. So I'm hoping to work on some of those things in the upcoming months. And one last thing is that, yes, I'm still setting up the HTTPS. That's the SSL Secure Site Certificate. And so for the time being, it's still a HTTP website. Some users may get a warning, a security warning, if they try to navigate there, but there's no actual security risk to doing that, and I hope to have that fixed within the next 1 or 2 upcoming weeks.
Great. Yeah, thanks, Jeff. Again, that's you've done a great job. It looks, the website looks a lot better. I think it's very operational, so it's a lot of work. And thanks for sticking with it. Okay, so next is we think we have some election results from Anya, and she was one of the members of the election committee, and I've been trying to also get hold of the new president. He might try to join us, you know, after you make these announcements. Jean-Francois, if you see that, I just sent you an email because he didn't register. So if you get a chance, we'll see if he replies here in the next few minutes and can join our meeting. So Anya, do you want to?
Sure. So the election committee was myself and Tim Collins, Adelaide Rhodes, Cynthia Saloma, and Ramon Rivera. None of them are here, I believe. So I have the honor of presenting the outcome here. We had a good voting turnout. We had a total of 108 votes. So thanks everybody for taking part in that. And with that, we can go over the results. So we only had a single nominee for president, and that was Todd Oakley, and he was supported by the vast majority of votes. So congratulations, Todd. Then the second officer position was the executive vice president for conferences, and we had two nominees, Vanessa Gonzalez and Inga Winkelmann, and the majority of votes went to Vanessa. So Vanessa is going to be the new VP for conferences. Then the third position was the VP for development and fundraising. And the only nominee was Joe. And again, he was confirmed by the vast majority of votes. So congratulations, Joe, you've been elected for that position. And then the fourth position was the executive vice president for early career development. We again only had a single nominee. That was Maria Carmen Ablan-Lagman, and she had the vast majority of votes in her favor as well. So she's going to be voted in on that position. For the, then we also voted for a student representative. We had three nominees, Jonathan Lawley, Kate Castellano, and Ricky Lee. So Kate is the only one who's here, I believe, of those three. And she had the most votes too. So congratulations, Kate, you are it. And yeah, that's all. So thanks everybody for voting. And only a few of the new officers are here right now, but congratulations to everybody.
Thank you. Yeah, and thanks for serving again and making this this process
I have
work.
emailed you and Kevin the report so you can preserve it for posterity.
Yes. Great. Congratulations, Kate, and all the other candidates. We will definitely work with you and anybody, all the new officers to make the transition. So that is great. Great news. Great. Thanks for the participation. Maybe Todd will join us here in a bit if he has time. I know he is. He's busy looking at some, working on some aquaria. Yeah. So the just a few other items here I just wanted to bring up, and this could be part of a discussion or going further. I don't want to take everybody's time this morning or this afternoon, but just a reminder that you have a GIGA bio project, and after seeing all the great genomes that are in the pipeline, it would be great to link those, your final results to the bio project. It's going to be posted on the website, newsletter. It's also linked to the EBP project as well. And they have standards in terms of the quality of their genomes. Some of ours may not make it because I know I have some draft genomes. And we also cover transcriptomes as well, because we first started the GIGA, we were including whole transcriptome assemblies as well as genome assemblies, and also just kind of putting together our standards. But feel free to link and refer to this bio project as we continue to add. And those that make the standards for EBP, they could also be included in that larger project. And then also kind of stemming from EBP and the Darwin Tree of Life, project is this Genomes on a Tree initiative, which is looking to add metagenomes, metadata to every genomic sequence and data set. So just wanted to bring that up to the community. If you're not aware of GOAT, that was discussed in the this year's Biodiversity Genomics 2021 conference. Always looking to add as much metadata to your genomes as possible. And this is a nice resource that fits right into EVP and then our project. So visit that if you get a chance. And I think that's going to really help out in terms of coordinating projects going forward. So that's pretty much all I have, besides any other topics that might have come up along the way today or the past few days. I had mentioned, for example, we had talked about DNA extractions earlier, and that just seems like it's an ongoing issue, and one that seems to be taxon dependent. The difficulty, I mentioned, we're part of the ASG project of Sanger, and their recent research report showed at least 30% to 40% failure of getting decent and high quality DNA extractions from diverse invertebrates.
Joe,
yeah.
Joe, before we go to that discussion, I think that on behalf of the membership and on behalf of the steering committee of GIGA, we want to thank you for your service. You've been the first, the funding president, the first real force driving this initiative, this community together from the times when we were not even a society. And this will not have happened without you. I'm very sure. There's a lot of other people who have contributed, especially, a lot of the Europeans have been driving a lot of this, but you've been a real force. Thank you for pushing us also to do things, and it's been great to work with you in this respect.
Well, thanks for everybody. Likewise, it's everybody. It's a community, so happy to be part of it. Support from all of you, because this is what makes it work. So hopefully we can just continue on. I'll be around again as a development and director here, so I'll continue to help out, and hopefully you and others will too. So thanks for that. Great community. So let's keep it. It's wonderful. It's a pleasure to work with you all. The whole board, Fabrizio, yourself, people who are not here. Love to see all the offshoots too, with Ignite, what Gert's done with that, and now Erga. And let's move on to we're going to need a lot of help if this is going to go out into scale. I mean, we'll see where we end up, but I like what it has become. And Todd mentioned, one of the reasons why he was going to join, and I don't want to put words in his mouth, but he liked it because it was a fairly laid back community. So, but we still get a lot accomplished. So anybody want to have any comments here in this open forum? Again, we'll just convey, Kevin's not here to record, but this is recorded. So if there's any comments here, we'll bring it forward to the next board meeting, whatever that convenes.
I'll just reiterate Joe, that I'm looking forward to the next meeting in person and see everyone. So I think Jean-Francois is still like pushing that, meeting in the Philippines perhaps. So that would be great. Jean-Francois, do you want to update? I mean, I know we discussed this yesterday, but maybe now that is the official meeting, do you want to tell us anything? What are the plans? Or?
Well, so the plan was pretty well advanced before everything was cut because of COVID. So there is this nice Philippine Genome Center, very beautiful building with some computer rooms. We can have some courses about genome assembly as part of the meeting. It's right in the center of Quezon City, which is the old capital of the Philippines, where actually Joe was born, if I remember well. And it's on the campus of the University of the Philippines. And we have a very good contact with Saloma Palomar, the head of the, Cynthia Saloma, the head of the PGC, one of the heads of the PGC. And I think we could very well reactivate this. We just need to find out if the situation gets better, COVID-like. So that could be, maybe it will be in the hand of the next conference director of GIGA, but I'm happy to pass the contacts on all the documents I've already gathered at that time. And I mean, for sure, we should plan on the next meeting on a place with some sun, sea, and lots of invertebrates waiting to be sequenced. And maybe by then, if the course I mentioned during the coffee pause, the course I'm organizing in Brussels works well, if we manage to get the genome done during that 1 week window, but we could imagine doing the same as part of the next GIGA meeting. Maybe we could have one community genome as part of the training for the young students taking part in the next GIGA meeting. That could be really fun. I think that Gert has raised a hand.
Yes, in between the Philippines, there may also be the opportunity to do what we have done here now with the Ignite Symposium, to have a GIGA Symposium at our final meeting for the Ignite program, which was supposed to happen in May this year, and it was postponed to November, and then we said we better cancel it, which was a wise decision. But we will have another supervisory board meeting in December, and there we will decide. We still aim to have an international conference before the end of the official end of Ignite, which is June next year. So let's cross fingers that the situation improves, and then we are more than happy, and we already have planned this. I've already discussed this, so we have discussed this with Joe, that we have a special day or half day or whatever there for GIGA, of course, and of course everyone is invited. And if it happens according to plan, then it will be south of Munich at the beautiful Lake Starnberg, with a beautiful view over the lake and towards the Alps. We just have to see where the pandemic brings us over winter, especially here in Germany.
Yeah, I hear it's not good where you are there, so hopefully it'll
we'll see. We'll see.
Get better by next year.
Facing a new wave these days. Yeah. Also in Belgium, they were talking about closing the schools again. They didn't do it finally, but it was a close call.
Gerd, actually next summer there is the Mollusk Congress in Munich, and a bunch of us in this talk will probably be there. So I don't know, maybe we could synchronize those things. And there is the Mollusk Congress, and then like a week later, also in Vienna, there is the invertebrate morphology meeting. So those might be opportunities to connect a lot of us.
Yes. Now that you say, I remember about the Mollusk Congress. Yes. Do you know actually when that is?
Yes, I'll tell you. Let me just check my calendar. Yeah, the Congress of Malacology, a little later than you said, is from July 31st until August 5th. And the International Congress for Invertebrate Morphology is from August 8th to August 12th. So yeah, maybe.
Yeah, now we will have to spend our money before the end of June.
Even under COVID, you think they won't give you an extension?
They have already given us 6 months.
Okay.
extension. I doubt they will. I will take this under consideration. And yes, now I also remember why I didn't have this on the radar, because it was too late in the year. But we will also have to decide amongst the Ignite program whether we want to have a final internal meeting or a big meeting. It all depends on, because we already have postponed it twice, and I don't really want to postpone it more. Yeah. But thanks for the reminder. I'll check that. Definitely check that.
There was an interesting remark by Chris in the chat about maybe we should do a poll to evaluate how people feel about flying long distance. And I think we should, in any case, I mean, in-person meeting are of course very nice, but now I think in the post-COVID area, there's no justification not to allow people to participate from afar. So I think we should think about doing the next meeting hybrid. I mean, if we can do it in person, but still allowing people to take part remotely. Now everybody is used to the new means such as Zoom. It should be not too difficult to allow remote participation. It was already planned actually when I was discussing with Cynthia Saloma for the GIGA meeting in the Philippines. We're already planning to make it hybrid, so, and that was before COVID. So I don't know how people feel about this.
Yeah, it's a tough one that should be discussed. I mean, definitely there's Zoom fatigue, and but it also has a higher outreach. And then I also understand hearing about how much a plane fare costs in terms of CO2 emissions. It's just it's incredible. So it's good to be green, and yeah. But thanks to you, Jean-Francois, again for setting this online meeting up on short notice. It's great. And the more meetings we have, the better. I mean, there's so much data, good results out there.
I still need to look in detail at the geographical distribution of the participants, but what I saw just by looking at the list, we already reached many countries. I mean, I know people from Visayas, Southern Philippines who participated, for instance. So, I mean, I guess online meetings or hybrid meetings are really more inclusive in terms of geographic participation. So we should really continue to foster such an international opening, I think.
Yeah. And I think with sorry, go ahead, Jeff.
I just thought, on the other hand, it's more difficult for students and researchers from those smaller international countries to fly over to the US than for, or Western Europe than for us to fly there. So that's also a consideration.
Yeah. And I know Jean-Francois has been, been thinking about a Philippine meeting for a while, and we have the resources there and contacts, especially with the new early career officer in Menchie. So it could happen. I think just some coordination, and yeah, if we fly there, I think that would give more opportunity to the local students who can't get around as easily. So
yeah, and
hopefully that'll happen in hybrids. Also, yes.
Right. One advantage of doing it hybrid also is in case of problem with COVID, it's easier to just cancel the physical part but keep the online meeting. It's a bit more flexible. So that's also more secure than basing everything on a physical meeting, and then if the frontiers get closed because of COVID, then you're all stuck and the meeting just dies.
Yeah. And one thing I've learned in this post, and I think we have as a board, is just trying to be inclusive and start listening more to the students and early career researchers. So Kate, I'm glad you're going to be on the board on that, and hopefully we can continue to improve, how to get early researchers and students more involved in this, like what Gert's done with Ignite. We just need more people with these skills. And you can get assistance from all around, again, from the whole community. You won't be alone in this. Just like the board here was served by both Nadej and Ksenia from Ignite, so they were both providing good input to us. Jeff, did you still have your hand up?
No, that was the only thing I said. I don't know why it's still up there.
So Gonzalo has to go. Again, thank you. One comment I was going to make that came up during the meeting was, we saw that a few genomes were duplicated or being produced by multiple labs, and that was one of the kind of areas that rationale for GIGA to be formed is to help coordinate sequencing, especially with limited resources. Now, I think it's okay to have duplicate or multiple versions of a genome, and I think they'll improve each other. And you can even contrast various methods or just having different and multiple replicates. So I thought that could be a topic here. But Jean-Francois, you have your hand up.
Yeah, I just had a small suggestion, also unrelated to what you just said, which was also very interesting. I was just wondering, maybe as a way to foster communication among GIGA members. So far we've been using Slack. The Slack channel has been not very active. So now many people who attend the conference went to Keybase to take part in discussion. So one possibility, if people think it's a good idea, would be to create, to actually move all these people registered to the GIGA4 meeting on Keybase, to register them into a GIGA team in Keybase, and to start keeping the role, and maybe phase out slowly the Slack, which is anyway not very active. And there are many advantages to that. In Keybase, all the messages get preserved. In Keybase, in Slack, after a while, all messages get deleted. And it's an open source, completely free platform. So I just wanted to throw this idea, and if people think it's a good idea, could very easily, as I said, just register all the people into a GIGA team.
Yeah, I'm fine with that. Now, actually, yeah, Slack got clunky for me. I just couldn't get my way out up there when I lost control of it. So if you want to help migrate people over, or just notify them that Keybase might be our main kind of a meeting point, and maybe with Fabrizio.
There's also automatic way of migrating people from Slack to Keybase. I saw a website about it. There's a way to import the people from one to the other one.
I don't want to throw spanners in the works, but I have heard from several people that they have severe security concerns with Keybase. And Keybase has been acquired by Zoom, and no one knows actually how long it will still be in existence. So that's but it's deciding between two evils, basically. I guess I've never understood what the advantage of Keybase is over Slack. Yeah, but why do we want to move to another platform?
One advantage I see is that Keybase is open source, so the code is actually there, and people who want can see the code. Slack is a commercial closed source initiative. I agree that
you also use Google Docs. It's also commercial. So, but I don't want to go into all these discussions. Do whatever you want. I just wanted to mention that people have raised. I know that some people do not use Keybase for Erga because of security concerns.
Okay, that's quite interesting. Yeah, I would love to know more about it. If you have some link to maybe some website talking about the security concerns.
Yes, I don't. Maybe it's all quite old, but there was a. I tried to figure it out.
Thanks. Yeah, it's an important discussion because we want to have a system that is peer-in, and yeah, so very important point, Gert. Thanks.
I liked how you were interfacing with Keybase when you were coordinating this meeting. Jean-Francois. So that seemed very, very well done. So anything that makes it easier to communicate, I think is the most important thing, with security, of course. Okay, is there anything in the chat? So are there anybody?
I just posted this link. It's from 2020, but I don't know how up to date it is.
Where did you?
I don't see your. Did you put it on chat?
I don't think you sent a link to everybody.
Oh, okay. Sorry. I sent it to someone else.
Thanks.
Not sure though if Slack is any better.
Right.
For sure, we need to think well about it. Yeah, I'll be
It
reading.
seems to have been working so far. There's just so many platforms, right? It's hard to keep up.
I'm easy. I'm using both. I just wanted to raise this concern because some people are concerned about security. And Chris already wrote in the chat that he can't. I think he can't use it in the Natural History Museum. That's something.
So this sounds more like setting at the institution. I mean, there are some ports that can be like with a firewall can be a bit tricky. I had one collaborator in Berlin had the same issue, and after he consulted with IT in his department, they just opened the port, and then it's been working fine with it. So it can be a setting, yeah.
So I like that idea you had, Jean-Francois, about bringing up a group genome, or I guess that would take a vote or something, but something that all students could work on. Or I mentioned the other day when Mark Blackster was talking about the cost of running PacBio, nanopore. Wouldn't it be nice if we just if GIGA had its own sequencer and somebody could run it and run our genomes on there? Because didn't sound, he was making it sound not very expensive, right? 5,000 to 10,000 a genome.
Talking about sequencing technologies, I mean, I just received some very interesting offers about the new Chinese platform, NB, I forgot the name exactly, something that works a bit like Illumina sequencing, but much cheaper. It looks quite interesting. I don't know if anybody ever tried it. They do the PCR, it's an emulsion PCR, like in the good old time of 454. So it is not on a glass slide like for Illumina. And apparently, according to what they say, it results in a lower bias towards GC-rich region, which could be interesting if you want to do some time like a quick and dirty whole genome assembly, fragmented, of course, just from this Illumina-like reads. Has anyone ever tried this Chinese machine? I'll be putting the name. I need to find the name. I'll put in the chat.
I think I've heard of that too. I can't think of the name right now. But is it much less expensive? What are some of the benefits?
Yeah, that's quite cheaper. I mean, like you can get. Okay, I forgot. I didn't do well the copy paste. I think you get like a 3. Let me see what they offer me. 30 gigabytes data per sample of paired N 150 for 120 euro per sample, and that includes library preparation. So that's quite interesting price, I guess. It's called DNBSec. I don't know why I always forgot that name. I will post it here. I don't know. I will be trying it this month. Normally we do some little trial, so I can let you know afterward if it works well. Yeah.
Yes. Hey, look who just joined us. Our new president. Hello, Todd.
Hello, how's it going?
Very well. Welcome.
Good. Welcome. We've just kind of having a free discussion right now. We've Anya, who left, announced all the winners of the election, and you were at the top of the roll. And thanks again for your willingness to serve as the next GIGA president.
Yeah, thank you. Running unopposed, so not too much of a surprise, but yeah, GIGA is something that I always supported, I always liked. I like the mission, I like the style, I like the collegiality, and so happy to try and help take it to the next few years. And yeah, my goal really is to grow in visibility and maybe size, but to keep the collegiality and the mission. So pretty simple, but happy to contribute. So thank you.
Great. Yeah, and you'll have Fabrizio and Gonzalo, Jeff, Kevin kind of around because they're we're on staggered appointments, and then of course we'll be around, and you'll have a lot of support, and that is a good thing about the community. Sorry you weren't able to attend this conference. I know we got busy schedules, but it's been recorded. We had a really great 3 days. And Jean-Francois, did you see that you said these recordings will be available for limited time? I guess what are people's feelings about having if you get consent from the speakers, you can start a channel, a YouTube channel, or have those posted somewhere, but at least they'll be around.
So right now the recorded are posted on the podcast platform of my university, which is it has some advantages. It's cannot be downloaded, so people can watch the talk but cannot download it. I wrote 2 months in the email just to give people a sense of urgency to watch the talks, because otherwise they think, okay, I'll watch it later, and 5 years after they haven't watched it. But actually there's no reason it would go out of phase. I can keep it forever. As long as I'm working at this university, I can keep it online there. So, I mean, I'm not so convinced of the advantage of doing a YouTube channel, but I could certainly, if there's a majority who likes to have it as YouTube, I could split the videos and make it online as YouTube on YouTube as well. That would be no problem. And yeah, also the website. So I use this open source platform called ScienceConf, and there's no reason also that it would turn off at any moment. So the website is probably going to stay live as long as we want. So I will be posting abstract book. I had no time to prepare it before the meeting. Anyway, I think also it keeps a bit the mystery about the presentation, not to have the abstract under the eye. So there will be a post-conference abstract booklet posted there. And also the picture we took together will be uploaded there as a group picture. So I think this can remain as a legacy of the GIGA4 meeting for many millions of years from now, hopefully.
Yeah, because I think GIGA3 is still up on Weebly. So
Yeah.
it's nice to have, and you can get the abstracts. So yeah, if you have the abstracts, the group photo, and
Actually, it would
links
be
to the
super nice
recordings.
to put
Would
back
be
online. Sorry, it would be super nice to put back online GIGA1 and GIGA2 as well, because they kind of they are missing online. One time I was looking for the dates of GIGA1 and GIGA2, and it took me a lot of time to figure out when exactly it was held because the website disappeared. It could be nice to keep them as a permanent record as well.
Okay, I can dig those up. Try to dig those up. Whatever we have from GIGA1. Gert probably has some archives of GIGA2, and yeah,
No
Todd was
videos.
at GIGA1 and
No
2,
videos though.
3.
No videos though. I don't know. I can't remember if we had.
I don't have
abstracts
videos.
back then. I think it was really informal. The meeting in Munich. I would have to dig it out. I can't remember.
I don't know if we had abstracts for GIGA1 either. I'd have to see.
I don't think so.
Even just a page with a group picture on the date. I mean, that's just as a, when people Google it, they see the series. I mean, when you will be at GIGA 20 in the future, it will be nice to be able to scroll back to GIGA 1.
Yeah.
And Todd, just before you arrived, we were also discussing about the idea of maybe switching from Slack to Keybase, and there was a bit of discussion about it. I don't know if you, as a new president, have any preference between the two platforms?
Slack to, what was the other one, did you say?
Keybase, that's the one that we have been using for the meeting here, actually.
I see. Yeah, I'm not informed on, I mean, we use Slack all the time, so I know that one and know it well, but I don't know the other one, so I could not compare.
Yeah, I will be sending you a little information about this if you want, and you can weigh in the way you prefer. There are pros or cons for both.
Sounds good, thank you.
Okay, I'm not sure I have much else to go over. Anybody else want to bring up any topics? It's again just great to have everybody together here these past few days, and hearing some really.
I may have missed, but is there a plan yet for 5, or is that not in the works yet?
Yeah, we were discussing about it also. So GIGA5, there are 2 ideas possible. Like one idea was to go back to Curaçao, where we have such a nice time for GIGA3. And the other idea was the initial plan for GIGA4 was in the Philippines, actually, at the Philippine Genome Center in Quezon City. So this is still in the drawers with plans already made with Cynthia Saloma, one of the heads of the PGC, so we could reactivate this plan as well. And we're also discussing about making it hybrid so that people who either cannot travel or don't want to add more CO2 into the atmosphere can take part remotely, whereas other people can be there at the same time physically.
Yeah, great. Sorry that I missed that discussion.
Don't worry.
This will all be recorded. You'll be able to see this discussion, and Anya and the election committee will send the results of the next board, so you'll see that.
So we still have 11 possible minutes of the meeting.
Go ahead.
Oh, sorry, the connection is a bit choppy. I was just saying, yeah, we still have 11 minutes till the official end of the meeting. So I don't know if anyone would like to bring some topics about the hurdles or future challenges of invertebrate genomics in the next few minutes, or otherwise we could as well close the meeting a bit earlier. That would be also perfectly fine.
I was going to say, Vanessa Gonzalez is going to be the next conference organizer. So
great. She's a collaborator of mine, so.
I maybe if I can take a word. When it comes to the DNA problem, the isolation, it might be worth probably to approach some of the companies who are producing those kits. I mean, we are not that big of a group, but given that there's millions of genomes to be sequenced, most of them being invertebrates, there's quite some revenue to make for some companies. And probably we shouldn't approach those big ones like QIAGEN or stuff, but maybe there's some startups that are interested to really go into the chemistry, help people to find out where the problems are. And like recently, one of my colleagues here in Munich, he worked with a startup in trying to get RNA depletion done in sponges. So they helped him to actually make baits to deplete those rRNAs to get metagenome sequences from sponges. And it works quite well in a good diversity of demo sponges. And this could be upscaled. But like for the DNA isolation, this might also be an option to find a company that is willing to put effort into this and that could also make money if we then in the long term probably sign up some contracts to use their product. Just an idea.
Good idea.
I mean,
we could even talk to the same company because it's in Munich and one of our ex-PhD students works there now. If we can convince him to continue that work in that direction, maybe they listen. But it's a good idea.
Yeah. Because at some point, I mean,
vertebrate
genomes are done, right? Pretty soon, actually. And then the big projects, they have to move over to the other stuff, and that's plenty.
Yeah, but don't forget the plants. The plants have proven also in ERGA very difficult, very difficult.
Yeah, a lot of genomic modifications to do still.
And of course, the big question is there a one-size-fits-all protocol that would work for every non-modern, non-vertebrate genome?
Certainly not, but at least on group levels it might work. I don't know. Or if they can go into the chemistry and identify the issues that we have, like Gonzalo mentioned earlier today about the blocking of nanopores and stuff, like to get rid of some chemicals that are still in there, some compounds that block the pores. I mean, I have really limited knowledge on chemistry, but those big companies or even smaller ones, they can help us, I hope.
And maybe a sponge kit, yeah, or a color kit, that would be pretty nice.
Yeah, stuff like this, or a crustacean kit or a Moldus kit. Yeah, I don't know.
No, a big market opening. Maybe Giga should make their own startup actually, and who generate funding for the whole association.
Yeah.
Yeah. Plus, having done a sequencer, this gives us good revenue then.
And with that revenue, we can fund some genome sequencing. So yeah.
Because the ASG is finding symbiodinium is giving a problem, and that's half of the genomes in there. That's surprising in a way. I think quantity is an issue, but there might be something there with the algae, as Ferran mentioned.
Yes, we had within ERGA, we had a couple of plants in the CBP sub-project, so the Catalan initiative for the Earth biogenome. And we had these issues with extracting with quality DNA, and then for the plants for which we had enough material and they were run through the nanopore, it was, I mean, the output was not enough, and so this, in fact, in the end, within ERGA, those pilot species that were including plants and algae, they had to be dropped or postponed, not to be included in the pilot. So.
Yeah, that's often something that surprised me. I have colleagues contacting me, okay, what's the recipe to sequence a genome nowadays? I'm like, well, it's still a bit, the dark art. You need to try many ways. And then people are very surprised that in 2021, there is not a one-size-fits-all approach to sequence a genome. And I mean, yeah, plants are a very good illustration of the headaches that can get in the way. So yeah, it's still an exciting, I think when it becomes too easy, it will not be fun anymore. It's still an exciting period for genomics now, because it's hard.
Any organism with polysaccharides, and I mean with crustacea, I know with copepods they also had issues with the nanopore, and this is something that it was in the literature decades ago, that you are always with a lot of methods you are co-extracting polysaccharides. So probably the chemist community, they might know, they might have some insights to contribute to the molecular biologist, but I don't know if through companies or through collaboration with molecular biologists more focused on the chemist part. But this is definitely something, as Michael was mentioning, that is I mean, it's something that needs really to be solved.
Yeah, something.
Yeah, and Kate mentions it's might be a good idea to have a session on DNA extractions, to test, what's been tried, any similarities.
And maybe a platform for sharing protocols that have succeeded. The people working on particular groups may have done some troubleshooting, and sharing that would be really useful, I think.
Yeah, it might be interesting to, if somewhere on the website there was a list of people involved in GIGA and what organisms they work on. So if someone working on a similar organism wants to contact you for information or help, that might be helpful, maybe.
Yeah, some nice molecule, some nice buffer I like a lot for everything slimy and full of mucopolysaccharides is cows, which has already been used for a while with corals, and it's based on guanidinium thiocyanate. And I just saw that now there is one commercial buffer that seems to be basically cows, maybe with some nice twist in it. It's called DNA RNA shield. And there's one advantage of it. I mean, when you do your cows yourself, depending on the pH, it's good for DNA or good for RNA. Apparently, this buffer, they found a magic formula that works for both. We're going to be trying it on a project very soon. I don't know if anyone ever tried this DNA RNA shield. It looks like if it works like a cow's buffer, so guanidinium thiocyanate, I expect that it will dissolve all the tissues and also dissolve any mucus or slimy stuff that cause problem. So, I mean, we'll be trying it soon. Maybe I can report at that time on the results.
And another thing that was brought up yesterday by Danielle was, this is not DNA extraction, but post-sequencing in the informatics, that it could take weeks, months to assemble, right? I mean, how are we going to deal with that going forward? And depends on the platform, and hopefully file sizes get smaller, but you still have to deal with having the hardware and resources to assemble. And we have huge genomes to deal with. Jeff. Or Nadej. Nadej, you're muted. Everybody's muted.
I was just waving to.
No, I'm
to
not
your...
longer muted.
Nadej?
No. Yeah, I can go. So yeah, I mean, it depends on the resources that you have, but I mean, for all genomes, the problems are not necessarily the same. Maybe if you have a genome that's big, but you can extract high quality DNA, then maybe you can do PacBio Hi-Fi, and with PacBio Hi-Fi, the resources I feel are less, are lower than for low accuracy long reads. And yeah, I don't think you have all the same problems at once. So I think you can work on that, but also you can aim for very efficient assemblers like WTDBG2, like Raven for low accuracy long reads. You can aim for Hi-Fi as an MDBG for Hi-Fi reads. A lot is feasible.
I may be coming to this point, so.
Well, thanks for the paper, Nish.
What?
Sorry, delay. Go ahead, Mish.
I mean, Nadej has shown yesterday that she has been trying lots of different assemblers and stuff, and Ramon Rivera, one of the students in Ignite, he set up a pipeline including all of these different assemblers for nanopore. For nanopore, it's like 7 different assemblers, combining them all in one pipeline to get the best possible assembly for whatever. So because for some species, one works better than the other, one assembler works better than the other. So you run them all at the same time. And in the end, you get one that is probably the best for your species. And he's now also bringing this to the PacBio platform. So this should be a one big solution pipeline where you just give in all the reads that you have. And in the end, you hopefully get the best assembly, including quality control and nice graphs and everything. So stay tuned. I hope Ramon is getting this done pretty soon. So most of the parts for nanopore is working already. So he's working on the PacBio part on it. And once it's there, then I think it's a good solution for everyone to play with and to start with, because then we have one reliable pipeline that can be reproduced and can be tested on multiple data sets, which helps to improve it, because yeah, with the input you get new ideas and you can tweak at some options and stuff.
That sounds very useful indeed. I just wonder, is there any step at the end to merge the results? Because that's really the tricky part, is to merge different assemblies to try to get a better one that combine everything.
That's the only thing that is missing. So it aims for the best based on some measures, like, I don't know, 7 or 10 measures that tells you, okay, based on these measures, this is the best. There's no merging included.
There is a bacterial genome assembler called Tricycler that does that exactly, like trying to merge different assemblies. Maybe the same approach could be implemented. I'll just be typing the name in the chat. Yeah, it's from the same author as a unicycler.
Yeah.
Ryan Wick.
Yeah, that could be put at the very
Sounds
end.
like we
but,
have.
yeah.
Sounds like we have lots of topics for future workshops. You know, like maybe a Giga assemblathon and the DNA extraction workshop. Be hands-on. It's just so much we could do here.
I see it's already 7:03. I don't know if there are some further topic people would like to bring. Otherwise, I would propose that we end here this nice meeting on time, about on time. Any further topic, hot discussion to be brought, or should we close it? I think I would like to thank everyone again for joining us. It was a really fun 3 days. I hope you all enjoyed it. And
yes, very much. Thank you, Jean-Francois.
Looking forward
See
to
everyone
again.
Bye-bye.
In the future. Take care. A great day.
Bye, everyone.
A nice weekend. Bye-bye.
Bye.
Cheers.
Have a good one.